Simon and I met a few years ago, and we shared a similar an interest in chaos theory, complex systems, and notions of order and disorder. So we hit it off straight away. So we're going to try and explain a project we did a few years ago and a new Wellcome Trust project we're working on called Cosmoscope, with recourse to the notion of what it means to collaborate as an artisan scientist, what types of new knowledge can be generated, and particularly Simon is going to talk first about what it meant for him and how it may have changed the nature of his research. And so over to you, Simon, and I will take over.
Okay, thanks for asking us down this evening. So I'm a physicist. I work at UCL. I'm basically interested in imaging cancer, so looking at new ways of diagnosing cancer, finding tumours, trying to characterise them, see if they're responding to therapy, that kind of thing. And magnetic resonance imaging is my main area of interest, MRI. So you've probably seen an MRI scanner before. It's this kind of thing, and you get these amazing 3D images of, in this case, a brain tumour. So this is the kind of thing that I'm trying to develop more of, basically. And I've got a particular interest in blood vessels. So tumours are quite interesting in the way that they develop blood vessels. There's a process called angiogenesis. So where you have a tumour forming, which is basically a bunch of cells, and they send out distress messages to the surrounding blood vessels to try and make them grow in and then sustain them. So we're developing ways of imaging this. With MRI, you can't see individual blood vessels. It's the kind of resolution you get, the size of the pixels are too big to be able to see individual blood vessels. But I'm quite interested in looking at much finer resolution images and then comparing them to the MRI to then try and work out what we're looking at. And these are examples of these kind of images. And they're really interesting, tumour blood vessels, I find. So normal blood vessels have a fairly well-constrained branching pattern. So you have a big vessel branch into smaller, 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 then comes back, joins back together again and gets bigger and bigger and bigger again in veins. But tumours kind of break all the rules. They have these really disorganised blood vessel networks. They're really inefficient. So one of the things you're often trying to do is inject as much drugs into tumours to try and kill them. But they develop these really poorly structured networks that mean you can't get drugs in particularly easily. So one of the projects I'm working on, semi off the back of the work I've done with Simeon, is to try and characterise how blood vessels within tumours, how well they deliver drugs to them. So this is an example of some of my data. This is using CT. This is looking at a tumour. And this is a tumour taken out of the body and looking at much finer resolution than you could look at if you were looking at it in the body. And you can see individual blood vessels, and you can see how kind of disorganised and chaotic they are. And this research led into my collaboration with Simeon. I call it collaboration rather than pets and owners. I don't know. It's a dynamic, Yeah. And this is the ultimate, the expression of the collaboration. This is Anakin in the organism. This was an installation in the cancer hospital just opposite my lab, actually, which I'm sure we could talk more about if we had more time. But this kind of explored the idea of complexity within tumours and their growth and how they responded to therapy, and it was meant to be a kind of educational tool for patients, as far as I understood it. So we started off by generating blood vessel networks. So this is an example that I generated looking at normal blood vessel networks and how these could then be disordered, how they could be sort of derailed by mutations and by things going wrong inside the body that causes a tumour. And you can see these much more complex and awful looking networks, but that are based on observations of real tumours. So this is kind of based on empirical evidence. And then we kind of developed a sort of dynamic process where we could grow normal-looking vessel trees like this one. This is kind of like kidney blood vessels, and then kind of mutate it and start growing something much more disordered, such as you see here. So it's like a tumour growing within a normal blood vessel network, and we can see that. So cutting to the chase, so this is how the kind of project developed, and we kind of took that further to the final installation, working with Nick as well, who's in the audience here. the key thing is how that then impacted on my work, because often it's kind of, I think the feeling is that there's a sort of imbalance perhaps between what the scientist gets from these relationships and what the artist gets from it. But I found actually that I got a lot from the relationship and the way we worked on the project and the kind of creativity that that kind of dealing with a fairly unconstrained project as you might have in art, whereas you're kind of in a much more kind of confined creativity as a scientist. Led me to kind of explore new avenues, I guess, with it as a scientist. And looking outside of kind of the standard MRI approaches and kind of research avenues, and bringing in new techniques like optical imaging. And this is kind of the most recent set of data that we've developed. This is looking at the entire blood vessel network within a tumour. And this kind of evolved naturally from kind of generating these fake blood vessel networks that kind of went into the installation, and then thinking more about how I could then use that in my actual research to look at real blood vessel networks. And what we're doing now is taking these complete blood vessel networks from tumours, we're simulating blood pressures and blood flow within them, and I don't think this is going to work, unfortunately. No. So trying to simulate what delivery of particular drugs would look like within these individual tumours. So these are real blood vessel networks. We can simulate blood flow using fluid dynamical modelling. We can then see what individual drugs would look like as they flowed through them, which you would see lighting up beautifully in this animation if it worked. And then comparing that back to real-world experiments. So we can predict how drugs are going to enter a tumour and then see how we can then guide future therapies. We're also looking at the growth of tumours. So as you saw, we had the kind of the network of normal-looking blood vessels and then how tumours grow within them. And we started developing models of what that looks like within breast tumours in this case. So you can see blood vessels forming into this tumour as it grows. And this is constrained by the kind of mechanical stresses and strains that you get within breast tissue. So all of this avenue, I'd say, came almost directly from the interactions between me and Simeon and kind of thinking in a kind of broader, more creative way about the phenomenon that I was interested in and kind of branching out from that. So, yeah, it was a positive thing all in all.
Thanks, Simon.
Okay.
Okay. So I will probably describe the same thing, but from my perspective. Same project, same sort of creative processes, same... So it came out of my sort of, when I was approaching Simon with this project, and also Nick Rothwell, the coder who's sitting over there, and also composer Rob Godman. It was like a collaboration between the four of us. It was really coming out of those notions of order and disorder. What does it mean to be ordered and what does it mean to be disordered? And this diagram here, very briefly, is it's a pity about the stripes, but it's a crystalline matrix with occasional things going wrong. Sometimes there's a node missing, sometimes there's an extra node. Very much what happens in nature, in solid-state physics. If you look at crystalline structures, minerals, metals at that level, they're never perfect. And one of the jobs is to try to find the perfect alloy for technological reasons. So this basic motif of order and disorder, very early in the project, I was talking with one of the professors of oncology, who's Mark Pemberton was his name, Professor Pemberton, and his eye happened to fall on this sketch, which I think I'd drawn in a sort of boring meeting at my university. And he said, oh, that's interesting, that's a classic diagram of tumour spreading through a tissue. And I said, no, it's not. It's one of my endless doodles, cartographic doodles of cities spreading into the countryside. So immediately I realised there's a kind of a relationship between different levels of description of different phenomena at different scales if you can get them into a similar diagrammatic form. It's a bit of a mouthful, but that became the basic leitmotif of the project. So moving right along to having cancer patients in my studio, doing sort of abstract drawings of their cancer. There were quite a lot of these. This was a drawing of the psychological ramifications, and this was a particularly affecting interview because this person had their lower jaw removed because they'd had bone cancer, and they were drawing the pain in their travel of the nerve pain that produces. So also I did this drawing of someone describing a cancer growing in her lung. So Simon sort of made a brief mention of branching systems, and that's something we both share, a fascination with L systems, Lindenmayer systems, which are an algorithmic way of generating plants and seems to have a kind of real sort of maps onto natural processes very universally. So I started to, just very quickly, investigate cancer imagery. So this is like drawings from a very simple cancer textbook showing a tumor developing. And so I've turned the cells into fields, and so I'm again conflating a sort of tumor topography with a cartographic topography. And so the cancer grows, eventually it turns into this huge tumor which has co-opted all the blood vessel networks and starts to metastasize, sending cancer cells through the blood vessel networks to colonize other areas. And I'm not saying this is some sort of analogy to the British Empire sending off its ships to colonize the rest of the world, but you could be forgiven for thinking that. And this, again, is an L system city with a sort of organizing principle governing most of the city, but this part here is a part of the city which is disorganized. It's a sort of anarchy within the city, not anarchy within the organism, which is doing its own thing, which is very much what cancers do. Don't draw any political inferences from that, or do. So anyway, this is what we've got here is this tree hierarchy that Simon and I sort of talked about and Simon generated through his software. And we've got cancers, or it could be mistletoe in a tree, developing at lower and lower levels on the branching hierarchy. And as it develops lower, you get a greater and greater sense of systemic collapse until you get a really awful situation. There's not really much hope for these things down here. And then I worked with Nick Rothwell to develop what in mathematical parlance are known as Voronoi networks. A Voronoi network is very simple. It's just if you see each person here in this room as a dot, just reduce everyone to a dot and then draw a line between the minimum distance between you and the next person to you and every other person in the room. We have a Voronoi network. It's a great savior when I'm on rush hour tube in the morning, thinking of everyone as a Voronoi network, I tell you. So we developed these cancer trees. So instead of the actual trunks branching, we've just got the points between the trunks, and it forms these nice cellular networks, which then led to the screen-based organisms, which grow and decay and get cancer, depending on the various versions that went on tour. And the software generated, we didn't know whether they were going to die or live or get cancer or be healthy, and they all just generated themselves over 15-minute cycles endlessly, and they still would be if we hadn't switched the computer off. So, and that's the window pattern. It was linking it to the system of the architecture screenshot, and I'm really racing through these images because I've got a few things to say afterwards. And then it went on tour. It transformed because it was also a piece of classical music written by Rob Godman, the composer. We took it to various venues and showed it in various kind of ways. We got extra funding from the Wellcome Trust to do these kind of vague music-human interaction, musician and screen machines, which played it in various places. And and it also went to the Huddersfield Contemporary Music Festival and King's Place and places like that as a chamber piece with this visual element. So it was a bit surprising that happened, but it was a good thing. So the basic, what I got out of it was a sense of, what I put in my little précis anyway, was that we tried to make it a genuine commingling of the scientific project and an artistic project. And I think I've worked with various scientists before, but with Simon, it was really the first time I'd worked with someone, and we really hit it off on a conceptual level, and were able to bring an artistic and scientific sensibility into a kind of single statement, and I found that really empowering because it allowed us to bring in the empiricism of science. It allowed the sort of empirical processes of the science that Simon was doing to be informed by perhaps a more rational or internal sense of imagining what might be, and also my sort of crazy artistic imaginings found a great sense of comfort in the kind of strictures and limitations placed by the necessity to actually make sure that all the knowledge, the information in there was actually scientifically respectable. So I think we managed to walk that razor's edge successfully. And so there's the various outings of it. And this, I suppose the point I was trying to make in this sort of coming together of a scientific method and an artistic method, I think it's the limits to the project which create the freedom to actually make something really interesting. I think you'd agree with that, wouldn't you, Simon?
Yeah, I guess I was touching on before about that kind of constrained creativity, you know.
Yes.
I guess that's what I feel scientific research is coming up with this idea, but within a very kind of constrained, universe, literally our universe.
The other thing that was interesting was that I think there was a symposium in which all the artistic collaborators and Simon and a lot of the patient groups who were involved came together. And I think what it did for Simon, correct me if I'm wrong, is it put you in touch with the kind of the ultimate audience or beneficiaries of your research. And it sort of humanized your research. I'm not saying you were being, I wasn't not suggesting you were being in any way inhuman before, but it did give you this direct, there was some quite sort of strong conversations. There's one person in particular who got quite upset at one point, accusing you of sort of removed, abstract...
Exactly, when you start talking about tumours, I mean, we do day-to-day the whole time in cancer and all these kind of things that actually are quite kind of taboos outside of the lab. So it's exactly that, they got quite irate, or at least one of them did, that I was kind of talking so freely about this stuff. Also,
I got into trouble quite early in the project because I was trying to sort of link cancer as a scientific phenomenon with other types of bad phenomena like earthquakes or tsunamis or natural eruptions on the Earth's biosphere where there's a lot of death and destruction. And I was trying to link the kind of disorder in the organism to the sort of planetary disorder, or to a cellular disorder. I was trying to look at various types of order and disorder at various scales, and in order to do that, you have to kind of zoom out. You have to, in a way, not dehumanize or not remove the felt affective elements of cancer, which has touched me, various family members, and many other people. But it allowed us to not necessarily explain cancer, but it allowed us to think about cancer in a more in a less sort of traumatized way. And initially, this didn't go down well with our patient groups or with the hierarchy of the hospital. But after a lot of meetings and a lot of explanation and seeing the final results and becoming involved in the artistic and scientific process, we feel that there was in the minds of the participants a new level of understanding about what cancer could be, and rather than seeing it as just some sort of bad luck or sort of dreadful thing, that there is actually a kind of an underlying set of causes and effects which cause cancer, and that it's just, I suppose in one sentence, the ultimate kind of reason for the existence of cancer we came up with was that in order for complex creatures to exist or complex systems to exist, in order for things to increase in complexity, and there has to be a lot of a whole process of mutation and change and sort of revolution, and these things aren't necessarily a kind of smooth, planar upswing. These are often cataclysmic, and so maybe cancer is the price we pay to be such marvelously complex organisms.